Every year, nearly 2 in every 100,000 people develop a debilitating neurological disease known as Amyotrophic Lateral Sclerosis, or Lou Gehrig's disease. The disease is characterized in patients by muscle weakness, impaired motor function, difficulty speaking, and respiratory compromise. Most patients die within 3 to 5 years of onset. Currently, riluzole is the only existing treatment for ALS. However, riluzole has been shown to only extend life by a few months and is largely ineffective in treating advanced cases. What we need is a novel and more advanced drug that can extend life by more than just a few months, maybe even a few years. So how do we do it? Before we answer that question, let's take a look at what causes the disease ALS. ALS is a disease that affects the upper motor neurons of the cerebral cortex and the lower motor neurons of the spinal cord. Degeneration of these motor neurons causes muscle atrophy and weakness throughout the body. 90% of all cases are sporadic and thus cannot be fully explained by just a single genetic mutation. However, the other 10% are familial and can be explained by a monogenic mutation. One such genetic cause of ALS is a mutation in the superoxide dismutase 1 or SOD1 family. SOD1 is a mitochondrial protein that protects the body from superoxide radicals that can cause oxidative damage in cells. The protein accomplishes this by converting superoxide radicals into molecular oxygen and hydrogen peroxide. However, in ALS, the SOD1 protein is mutated and clumps together to form large aggregates of protein. These aggregates can be harmful to many important processes of the cell. For example, protein function can be lost when mutant SOD1 co-aggregates with other proteins in the cell. Proteasomes, which are responsible for normal protein degradation in the cell, can get clogged and rendered inactive. This disrupts the regulated degradation of misfolded and unneeded proteins in the cell and can lead to induction of apoptosis or programmed cell death. And last but not least, normal mitochondrial activity like oxidative phosphorylation and ATP production can be disrupted, resulting in cell dysfunction and death. If we could find a compound that reduces toxicity from mutant SOD1 and increases motor neuron survival, we could be on our way to finding an effective therapeutic. How do we develop a new drug to better treat patients? From the lab to the clinic, finding therapeutics for amyotrophic lateral sclerosis. A new innovative method to discover novel and effective therapeutics for difficult diseases like ALS is high-throughput screening. In a high-throughput screen, a large library of compounds is tested on a specific cell type to identify drugs that produce a particular phenotype. Before you can begin the chemical screen, you must first build a library of compounds to test on your cells. Chemical libraries can consist of anywhere between 10,000 to 1,000,000 different compounds. The more compounds in your chemical library and the more diverse their structure and function, the higher your probability of identifying hit compounds that induce the desired phenotype in your cells. You also want to determine the types of cells you will test your compounds on. Because you're dealing with ALS, a disease in which SOD1 toxicity specifically harms the lower and upper motor neurons of the brain, you want to test your compounds on cultured motor neurons. In this case, you will test your compounds on wild-type motor neurons and mutant SOD1 motor neurons. Why? The wild-type motor neurons will allow you to test whether the compounds can improve survival of sporadic cases of ALS, and the mutant SOD1 motor neurons will allow you to test whether the compounds can improve survival of familial cases of ALS. To grow motor neurons, embryonic stem cells are first co-cultured with feeder cells, which secrete important factors that maintain the undifferentiated state of the ES cells. Next, these feeder cells are removed from the culture and the ES cells are allowed to differentiate into 3D structures called embroid bodies. Retinoic acid and sonic hedgehog, which play an important role in early growth and development in mammals, are then added to the culture to differentiate the stem cells of the embroid body into motor neurons. These motor neurons are then dissociated and plated on 384 well plates with trophic factors that promote the development and survival of these cells. After the motor neurons have been left in trophic factor-rich culture for 4 days, the trophic factors are removed and candidate compounds are added. Compounds that work to improve motor neuron survival will prevent cell death in the absence of trophic factors. The motor neurons are left in compound-treated media for another 3 days and then imaged using an imaging machine. Motor neuron survival is calculated using the software and hit compounds are identified. The screen is repeated with more stringent conditions and a select number of hit compounds are identified. High-throughput chemical screens can help us identify novel compounds that support motor neuron survival. These compounds can then potentially serve as effective therapeutics for patients with neurodegenerative diseases like ALS. By taking research from lab to the clinic, we can not just help science, we can help patients.